part of any program has to be identifications of patients that can receive this therapy. That can come from within your own group or from referrals from the outside. So if patients are referred from the outside, then the program has to be able to identify whether these patients are even indeed eligible for the treatment, clear the insurance as well as determine medical necessity, and then eventually get the patient in the door and medically evaluated to determine whether indeed the treatment can be successfully done. So this does require setting up a robust process to be able to handle everything from insurance referrals and contract negotiations with a wide variety of insurers to then a medical team that can actually quickly evaluate these patients and move on to collection of cells and treatment. One of the major issues that we're facing in this is that patients can't wait weeks to be able to get this therapy. And with those patients with, for example, diffuse large B-cell lymphoma that's refractory to prior therapy may only have weeks to months to live, and time is of the essence, and it's critically important that we try to get through all these stages and steps as quickly as possible. Once you have identified a patient that's appropriate to go forward, then the first step is to actually collect the cells by apheresis or leukapheresis. And this depends a little bit on which manufacturer you're going to go with, as each manufacturer has different requirements for shipping the cells, either fresh or cryopreserved, from the collection center to the manufacturing center. And so this requires careful coordination between your intake group and scheduling group and the manufacturer. And it usually requires of 3 to 5 days in advance to get the products, the timing set up. Once the cells are collected, you may also need to make sure that there is a appropriate either vein or vein-to-vein collection, or it may require a central catheter. This obviously requires thinking in advance to get the line placed and to be able to manage their collection. Once the cells are collected, then your cell collection facility will then ship the cells directly to the manufacturer or cryopreserve them. Obviously, the chain of identity and chain of custody is absolutely critically important, that all the procedures are followed to ensure that the sample will return to the same patient. These are patient-specific treatments, and that's much of what the site requirement is from the manufacturers and from FACT accreditation to make sure that those processes are all in place. So once the cells are collected and shipped off, then we have to monitor the patients until those cells, the anticipated date of return, and obviously it's critically dependent on how fast the manufacturers can make the cells and how reliable they can do it. Once patients' cells have been collected and shipped off to the manufacturer, we're often faced with trying to control the lymphoma or the disease while we're waiting for those cells to come back. And this really depends on how quick the manufacturer can make the cells in some facts and the aggressiveness of the disease. Most of these patients have relapsed refractory disease and are in desperate need of therapy. And unlike in the clinical trials where often bridging therapy was not allowed, we often are faced with having to give bridging therapy to try to keep people alive and symptom-free to control the disease while we're waiting for the cells to come back. Once those cells are back, then we can schedule lymphodepletion chemotherapy and to move on. But many patients will require bridging therapy, at least at this stage of the new development of these drugs, because patients are often referred after nothing else is successful. I'm hopeful that we'll be moving to a more earlier stage in treatment where patients will have less disease burden at the time, and that'll likely make the treatments more successful and less toxic.